Hello. How are you, everybody? Today, I would like to present for you one paper which is on the virome of the mosquito, which was done on Guadeloupe. So the title of the paper says stable, distinct core eukaryotic virome in different mosquito species from the Guadeloupe using a single mosquito virus metagenomic. So the method is by using a viral metagenomic. So we'll see the virus. So maybe for today we'll see only the methodology part. So to have a look as an introduction, it's good, to have a better understanding of the commensal viromes in case of the mosquito species in the study area, because this is very important to have a better assessment of the mosquito-borne disease risks. And also, it is very important for the vector competency, and that finally will provide an enlightenment on the mosquito controls. Therefore, having a bit to have a better understanding of the commensal viroma of a mosquito is really very important in order to control such mosquito-borne diseases. So therefore, the objective of this study is performed with a viral metagenomic sequencing on the individual and as well as on the pooled Aedes aegypti and the colex, which is collected in the study area. And then after, there was a comparative analysis of the eukaryotic environment and the phagosome were connected between the gender and the location of the mosquito species. So there are the gender, either the male and the females, and also the location in different places, but also the mosquito species with the Aedes aegypti and also the Culex. So therefore, there was a kind of comparative analysis of the eukaryotic environment. And finally, the several novel viruses were identified with this study, and subsequently they have used for phylogenetic analysis and also a QRT PCR analysis in order to investigate the possible core viruses in the mosquito population. And finally, the correlation analysis was used to identify the relationship between the phage coating and the bacterial marker genus. So let's have a look, how they collect, the mosquito and also how the pooling was done. So the first thing what they do is that they collect the Aedes aegypti and also the Culex were collected. Once they have collected, the mosquito species were determined for morphological identification by using a binocular loop at a magnification of 56 using the morphological description for each species. So what they do is that once they are collected, they go for morphological identification of the species, and also they store it in minus 80 until processing. And then later, there was a kind of a total number of 95 mosquitoes were sampled, and they group into four pools for sequencing. So one is in male and female of the Aegypti and the Culex, right? So totally male and female two for both species. Totally there was a four pool for the sequencing. So therefore, the sequence is six pools for each species and the gender and sampling location. So five pool with a single mosquito, right? And one pool containing five mosquitoes. So for one pool, there was five mosquitoes. So in total, there was a kind of 36 pools. And so therefore, as a negative control, there was PBS for it. So how is the sample processing and sequencing? The sample processing and sequencing were it is an optimized sample preparation protocol for a viral genomic technique called a NATO view was used to analyze the mosquito pools in the individuals as well as the negative control. So therefore, briefly, the whole mosquito were homogenized, right? The whole mosquito were homogenized with 200 microliter PBS in the mini lysis tissue homogenizer for 1 minute at 3000 RPM using 2.8 NMCL microbead and centrifuged for 3 minutes. And then after 150 microliters of the supernatant were used for filtration by using 0.8 microporcite to enrich for a viral particle. So now we are in the filtration step. Then the filter will then treated with a cocktail of the benzonates and the micrococal of the nuclease in a homemade buffer. With that, it was treated, and the objective of this treatment was to digest another free floating nucleic acid. Obviously, there might be some nucleic acid which are not floating, so to digest that one, we use another the benzonates and the micrococcales nuclease to digest. And then after RNA and DNA were extracted by using a huge and wire RNA mini kit and the reverse transcribing random length using the slightly modified whole more kit was procedure was used. So the W2 partook were purified and the library were prepared and for Illumina sequencing using the extra and library preparation that's enough for Illumina and then a clean up after a library synthesis was performed and sequencing of samples performed is an extract 500 highest throughput platform for 300 cycles. That means 2 times base pair index. So for next time, we'll see the result of this study. Thank you for watching.